Welcome to the MaAsLin2 tutorial, a powerful tool designed to identify microbial associations with metadata features. MaAsLin2 has been seamlessly integrated into the CosmosID-HUB, allowing you to access its functionality through an intuitive graphical interface without needing to navigate complex command lines. MaAsLin2 requires an abundance table of taxa or functions and metadata. Your metadata should include all the variables or covariates that you want to test against your microbial data. To demonstrate the MaAsLin2 tool, we'll be using an example dataset of chicken cecal microbiome during salmonella infection. Once your data is analyzed and your metadata is input, simply select all of your samples and click Create CA. Begin by naming your comparative. You can then select the MaAsLin2 comparative for either your taxonomic or your functional results by selecting the appropriate comparative workflow. We recommend running MaAsLin2 as a separate comparative, and cohort setups to the left should be ignored. Here you'll see a number of workflow parameters, such as the filter set, which is total and filtered calls, the metric, relative abundance, genome normalized reads frequency, and raw reads frequency, as well as the taxonomic level by which you want to conduct your analysis. Next is choosing the fixed and random effects, which is critical for the success of the MaAsLin2 analysis. The fixed effect variables define how samples are grouped into comparative cohorts. You can add multiple fixed effect variables, such as infected or uninfected with Salmonella enterica for this example. You can select the data type, such as numeric or categorical with a reference level if you have a controlled factor, such as an uninfected or control group. If you select categorical without a reference variable, it will assign the first alphabetical metadata variable as the reference variable. For this example, we'll select the uninfected as the reference value. You can also add multiple fixed effects if you want to compare multiple metadata variables. For this example, we want to compare days post-hatching to look at the effect of the day of hatching and age of the chicken on their microbiome profile. So we will add this as a fixed effect. While there are no random effect metadata collected for this study, you can add random effects such as BMI, age, or sample ID for longitudinal studies. These will build into the model to account for potential confounding factors that may introduce noise, such as subject-specific differences. Next, you'll find other parameters, such as the minimum abundance threshold, which sets the number for a minimal abundance required for a feature to be included in the analysis. This helps to filter out low abundant noise, and we recommend 0.001 as a default. Higher thresholds may exclude noise, but could also miss low abundant features of interest. The minimum prevalence percentage defines the minimum proportion of samples in which a feature must be present to be included in analysis. Typically, a threshold of 5 to 10% is used, which means that 95% of samples must have the detected microbe in their microbiome profile, but this can be adjusted based on study and research questions. The Q-value significance threshold controls the maximum acceptable false discovery rate, or FDR, for multiple comparisons, and this helps to reduce the risk of false positives. A common range is between 0.05 and 0.25, with lower values for more stringent control. We recommend 0.1 as a recommended value. Normalization accounts for differences in sequencing depth and compositional biases across samples. By default, and for most applications, we recommend TSS or total sum scaling. Definitions for the other normalization methods can be found in our documentation. Transformation applies a mathematical transformation to the data to meet assumptions of statistical models or reduce skewedness. For most applications, logarithmic transformations are most applicable, but others can be selected based on your study questions. The analysis method determines the statistical model used to test associations between features and metadata variables. For most applications, and our default recommendation, we recommend the linear model. Other more conservative models are those that are better for zero-inflated continuous data can be selected according to your study design. To learn more about these analysis methods, you can consult our documentation or the MaAsLin2 manuscript. The last option is the multiple hypothesis correction method. We recommend the Benjamini-Hochberg procedure due to its balance of sensitivity and specificity. This controls the false discovery rate, which is expected proportion of false positives among the declared significant results. It's preferred for large-scale studies with many comparisons, and it balances false positives and false discovery rate effectively. Other methods can be used for smaller data sets or others with many dependent hypotheses. And definitions about these multiple hypothesis correction methods can be found in our documentation. If any numeric variables are selected, you can standardize them by applying a z-score standardization, which transforms continuous metadata to have a mean of 0 and a standard deviation of 1. You can also select the number of features that are included in your heat map, as well as the version of the workflow by which you want to compare. If running functional analysis, there are other options as well, such as the functional type, such as metapsych pathways and enzyme commission hits, and other metrics such as relative abundance and copies per million. Fixed and random effects must also be selected for this particular example. And we have a recommended default parameters that can be used according to your model. Once everything is set, we can start the analysis by selecting Create. This process may take a few minutes to complete depending on the complexity of your model, but you can monitor the status and view the comparative in the comparative analysis menu. Here we'll navigate to the comparative analysis that we just generated. You'll find a table of your input data and your input metadata, as well as the association results. The results table is where you'll find the most detailed statistic of each feature, including p-values, q-values or false discovery rates, and the effect size, as well as which taxa were enriched in which cohort. Visualizations are also available to aid in interpretation. The heat map shows the associations between features and metadata variables, where deeper colors indicate stronger associations. The volcano plots show negative and positive associations based on your metadata category. The color coordination is based on effect size and q-value. Forest plots can help visually summarize the significant result features and their effect sizes from the model contrast along with their confidence intervals. The x-axis represents the contrast effect size, with the y-axis representing the significant features that were found in respective contrast of interest. Lastly, you can view individual abundance box plots for each of the significant taxa by exporting the data as a zip file. Within this zip file, you'll find a variety of tables and visualizations, as well as tool logs and all the visualizations in PDF format. Box plots will be available in the hub on the next iteration of the MaAsLin2 release. A few tips to get the best results from your MaAsLin2 analysis is ensuring your metadata is clean and consistent, using appropriate normalization techniques, and always validating your findings with biological replicates or independent datasets. And make sure to avoid overfitting by limiting the number of covariates and use proper statistical adjustments. Thank you for joining this tutorial. For more detailed guidance, be sure to check out the comprehensive documentation available in the CosmosID-HUB. You can find it by navigating to the documentation menu and find a full descriptive overview of the tool with screenshots and parameter definitions, as well as a table of recommended workflow parameters. Should you have any questions about the tool or issues with the software, please give us a shout at support@cosmosid.com, and we'll be happy to help you as you analyze and unlock your microbiome.